Well, thank you all for joining today for the third of our Northeast IPM webinar series, third out of 4. So today we'll be talking about biocontrols, the efficacy costs, and also include a bit on organic production in there. So we're going to start off today with Amara Dunn-Silver, who will focus on the basics of what actually is a biocontrol product, and also will show some resources to help us learn more about biocontrols. Generally, I wanted to point out before we get started with Amara's presentation that she's got a lot of links to share with you guys, and I'll stick those in the follow-up email. So don't feel like you have to desperately be writing those as we go along. We might also stick them in the chat, I guess, if we feel like it. But we can definitely stick that in the follow-up email. So you'll all be getting that. Yeah, so Amara is a biocontrol specialist for New York State IPM, and she studies how best to implement biocontrol strategies across all commodity crops, not just specific to fruits, but she knows a lot about fruits. So she's going to be today more talking generally to us about the biocontrol products, and then we'll delve into the nitty-gritty with the next 2 talks. So that looks great. Amara, I can see your slide and you can go ahead.
So I'm going to see if I can move the Zoom things out of the way so I can see what's going on. And hopefully my screen is still there. It's
Yes.
always exciting when Zoom works. All right.
Everything
So I like to let folks know when I start these talks that I consider myself someone who is experting in biocontrol. That is, I devote a lot of time to learning about biocontrol and I practice engaging with the topic a lot, but I certainly don't know everything there is to know about biocontrol. So I'm sure that I'm going to learn things here today. And as Janet mentioned, I don't work exclusively on fruit, and so I'm not even sure I would say that I'm someone who experts in fruit. So you all know a lot more about tree fruit production than I do. So the things that we'll talk about today is I wanted to spend at least a little bit of time on these terms: biopesticides, biocontrols, biostimulants, answer any questions about these that you might have. And then we're going to talk a little bit about how biocontrols work, or modes of action, and interspersed throughout these topics, and then also a little bit at the end, I wanted to highlight some resources, at least from the New York State Integrated Pest Management Program, that may be relevant here. There are probably many other great resources out there as well. This isn't meant to be an exhaustive list of all the resources. I'm told that you all feel comfortable interrupting and asking questions, so please continue to feel comfortable doing that, whether you want to unmute yourself and ask. We're a pretty small group, or put something in the chat. I'm happy to stop and address questions as we go. All right, so I'm hoping for some honest answers, and you can respond any way you want to this. How comfortable do you all feel explaining the differences among these terms to growers? And if you are comfortable with the reaction options in Zoom, you can do some thumbs up, thumbs down. I'm not sure. I thought about putting the scream emoji in here, but I decided no on the scream emoji. Or if you have your video on and you want to show your reactions that way. I'm just kind of curious how folks feel about this. Seeing 1 thumbs up, 2 thumbs up, 1 wiggly thumbs up. Okay. All right. So one of the ways that I like to think about this is with this Venn diagram. If it doesn't resonate with you, that's okay. If we think about things that control pests, like everything that controls pests as this green circle, the overlap between living things, so insects or other arthropods, nematodes and microbes, and things that control pests, that's where biocontrol happens. And sometimes you'll hear the term natural enemy or biocontrol agent, living things that are doing biocontrol. Now, if it's a microbe, a bacteria, fungus, or a virus, it's considered a biopesticide, and it's regulated as a pesticide. Also, plant extracts and biochemicals are considered biopesticides, things like potassium phosphate or other things in that kind of family. The decision was made, I don't know when, a long time ago, that if it's an insect or a nematode, those living things are not regulated as pesticides. They're exempt under the Federal Insecticide, Fungicide, and Rodenticide Act. So the microbial biocontrol agents still biocontrol, but biopesticides regulated as pesticides. Biostimulants are kind of off on their own here because some of them are microbes, So that's a common thing with biopesticides, but their purpose is not to control pests. Their purpose is to improve the health of plants or the quality or access to nutrients in the soil or something like that. So that's how I like to think about the difference between these bio terms: biocontrol, biopesticide, biostimulant. Folks sometimes ask me, well, how do I know if it's a biopesticide? If it contains a microbe or a plant extract, and those are pretty easy to recognize as an active ingredient on a label, and its purpose is to control pests, then it's a biopesticide. The EPA registration number is another good thing to look for if you're trying to figure out biopesticide or biostimulant. However, I have an asterisk here, and that's related to minimum risk pesticides. So is everybody familiar with minimum risk pesticides? How do we feel about these? So these are pesticides that contain only active ingredients that are on particular lists that are at least currently allowed by federal regulations, things like thyme oil or citric acid. As of right now, they're not currently registered with the EPA or the New York State DEC. I'm not sure about other state agencies that regulate pesticides. And so they don't go through that testing process that they would if it were a regular pesticide. Sometimes the labels say that they're exempt under FIFRA, and they'll often mention 25B because that's the relevant section, but they don't have to have that label. And so when I am talking to growers and gardeners, I like to remind people a pesticide is any substance or mixture of substances intended for preventing, destroying, repelling, or mitigating any pest. And so the EPA considers microbes, plant extracts as substances, but insects and nematodes not. However, I often like to remind folks, sometimes people see the bio part of biopesticide and they think, oh, well, it must be safe. There's no risks. I can use it any way I want. And of course, this isn't the case. You probably all know this. So they have restricted entry intervals. They have required PPE. Some of them are approved for organic use, but not all of them. And some of them have some real environmental hazards. So this is an excerpt from a biopesticide label that can be harmful to bees and pollinating insects. So I like to remind growers about this, remind gardeners about this, that they should still be reading and following labels. Related to this section, if you're interested in learning more, reading more about minimum risk pesticides, or someone asks you about minimum risk pesticides, the New York State IPM program has a page where we list the active ingredients that are eligible for use in these pesticides. And there's also a fact sheet about nearly all of them. There's a new one that's been added since the fact sheets were made that has information to the extent that could be found about whether those active ingredients are likely to be effective. All right, so that's what I have to say about those terms: biopesticide, biocontrol, biostimulant. Any questions so far?
So would you include SARs as biostimulants? The resistance-inducing materials like Blossom Protect,
It depends.
double nickel, etc.
So double nickel is for sure because the active ingredient is a microbe. I think Blossom Protect is also a microbe. But for example, I think methyl salicylate based products, that active ingredient is not considered a biopesticide, I'm pretty sure. So it depends on what the active ingredient is. It's less about the mode of action and more about the active ingredient from a technical standpoint in terms of what the EPA considers a biopesticide and not. And there's actually a website, I didn't include the link, where the EPA has a list of all the active ingredients that are considered biopesticides. From a grower standpoint, I think it's probably more important to know, is it a pesticide or not a pesticide, than to know if it's a biopesticide versus a pesticide. All right, so let's talk a little bit about how biopesticides work. So some biopesticide modes of action are very familiar or very similar to conventional chemical pesticides. So these are my attempts to draw cartoons of an insect being poisoned by a biopesticide, or a biopesticide applied to a leaf, and either the living microbe or something it produces or a plant extract is killing a plant pathogen. That's pretty similar to how conventional pesticides work. You spray it, it kills the pest. Especially folks who work in tree fruit are also very familiar with pheromones. So the pheromones are also biopesticides. That's pretty common, pretty normal. Some of the modes of action for bioinsecticides are also pretty familiar. So we have conventional pesticides that are insect growth regulators, and there are some biopesticides that will also stop the growth of insects, which I've attempted to diagram here. There are some biopesticides that repel insects, and there are some that cause insects to stop feeding. And then there's some other biopesticide modes of action that are a little bit less familiar. So here we have a blue fungus that's killing an insect, or sometimes these fungi eat other fungi. And I think of this as being different from poison because the fungus is actually colonizing the pest fungus or the pest insect, and killing it as it colonizes it. We also have biopesticides. These would be live microbes that colonize the surface of a plant, and they exclude the pest from that plant surface, whether it's a leaf or a root. There's no space for the pathogen to land and colonize. And then this is my attempt to illustrate SAR, or inducing the resistance of a plant. And there are some non-biopesticides that work this way as well, but it's a mode of action that's more common, I think, with some of the biopesticides. And then this last mode of action is complicated. So this is my attempt to depict the fact that some biopesticides work by growing a stronger plant, which probably sounds a lot like a biostimulant, right? Biostimulants grow healthier plants. And we know that healthier plants are often more resilient when they're attacked by a pest. And it just comes down to what the label is claiming. So if the label is claiming that the growing of a stronger plant is for the purpose of pest control, it's a biopesticide. If it's just to grow a stronger plant, then it's a biostimulant. I put more information about these modes of action into 2 blog posts. So there's one that focuses on biopesticides that target plant diseases, and there's one that focuses on biopesticides that target insect pests or arthropod pests. And those links will be in the email that Janet sends out as well. I think why the modes of action matter so much for biopesticides is that there are really important implications for when you might use the biopesticide, and also what success looks like. So obviously, if you have a biopesticide that is a living microbe and you need it to colonize the surface of the plant before a pathogen shows up, you have to get it on the plant before the pathogen shows up, and you have to be mindful of how long it's going to survive on the plant. Is it going to be washed off in rain? When do you have to reapply it? And then the what success looks like piece, I think, can be really important, especially for some of the bioinsecticides. So some of these bioinsecticides will stop insect feeding within a day or two, but it might take a week for the insect to actually die. And so I think it's important to be clear about what it will look like if the biopesticide is actually working. And then this is connected to modes of action, but not a one-to-one connection. If it's a living microbe that is the active ingredient in the biopesticide, then there's a whole slew of other questions that have to do with what's the shelf life of the product, either before or after opening the package, how should it be stored, and optimal conditions for application, whether that's what sort of temperature or amount of sunshine is happening in the field, or things like pH of the tank when you're mixing it up. So a resource from New York State IPM to know about this, I've started creating these biopesticide profiles to focus on specific biopesticides. So they're around a product, not around an active ingredient. They have some information that could also be found on the labels, like an REI is on there, but they're certainly not meant to replace a label. what I did was I looked at a lot of different sources, EPA registration documents, literature from the manufacturer, pesticide labels, sometimes looking into some scientific literature, and trying to put into a really short, quick to read format, practical information that a grower might need if they're wanting to use a product and they need information about how they can use it most effectively. So things like how long it lasts on the foliage in terms of UV tolerance or storage or shelf life, things like that. And this link will be in your email as well. All right. I had a couple more resources to share. Any more questions about the modes of action for now?
Well, do you consider BT toxin as a biocontrol or a biopesticide?
So I think I would consider BT toxin a biocontrol and a biopesticide. They're not mutually exclusive. I think where you would have something that is so partly BT is a biopesticide because the EPA says it's a biopesticide. And that's a piece I neglected to mention. Actually, they call them plant incorporated protectants. So if a genetically modified plant has genetic material from a microbe that controls the pest, that's regulated as a biopesticide too. So BT is definitely a biopesticide. I think a lot of biopesticides are also biocontrols. Definitely all the microbial biopesticides I would consider biocontrols. It's a living organism or its product that is controlling the pest. The biochemical biopesticides, I don't usually think about as being biocontrol. And also I recognize that it doesn't really matter to a grower. Probably in most cases, something like, is it OMRI approved? Or, is it a pesticide or not a pesticide? I think those are probably more important questions to a grower.
Yeah, I get a little bit antsy when people say BT, dipel, et cetera, is a biocontrol, because it kind of people use that to say, well, I'm not using a pesticide. Well, you are using a pesticide. It's not even acting as a living organism at that point. It's just another form of chemical control. I mean, it's good stuff. I'm not against it. I just think it gets kind of over biosized, But anyway, did you say that pheromones are considered biopesticides?
Yes.
So they have an EPA number, like cobbing moth pheromone?
I believe so, yeah.
Okay.
Someone can correct me if I'm wrong.
No, that's correct. That the pheromones will be biopesticides.
And I think your point about someone saying, oh, I'm using BT, it's not a pesticide, you're absolutely right. That's an incorrect statement because the BT is definitely a pesticide. And you're absolutely right that it's easy for people to think, oh, it has the word bio in it. It's safe. It's, it's all these things. It's not a pesticide. And I think it's important to correct those misconceptions. I feel that biopesticides, when they're based on a microbe, when they're based on a plant, they're also biocontrol. And if I don't know if you've heard Dan Wickstead from the pesticide safety education program at Cornell, but he'll tell you any pest management strategy has a risk, right? I mean, he'll explain to you all of the ways you can hurt your back by hand weeding, right? Relative compared to the risks of using a chemical herbicide. So I think it is important to be clear with folks that we're not talking about things that are no risk. We're talking about sometimes different risks, sometimes lower risks, and to have clear language around that.
Yeah, and especially in the case, and I don't want to beat this to death, but especially in the case of BT, it's not functioning as a microbial infection process. The BT, the bacteria is not growing inside the host. That may happen a little bit, but really it's just a chemical toxin that's disrupting the gut lining. So it's really acting as a chemical at that point. But anyway, let's continue. I don't want
to. So yeah, one of the other resources I wanted to share with you, and I'm not going to talk about efficacy today, but I have been spending time trying to pull together efficacy data on biopesticides because that's the biggest question people ask, is they want to know if it works. And I am not able to do all of those trials, especially not for all of the crops. So I pull together what are basically giant spreadsheets where I have a row that summarizes each product in all the trials that I can find. And, tries to put in one place some basic information about did it work, and then some of the details so that you can assess, like, what does it mean if it looks like it worked. My hope here is that this is a tool that is useful to faculty and extension educators. A lot of growers are not going to be interested in a giant spreadsheet, but you can download this spreadsheet from our website, and then you can sort by anything you want. So you could sort it by product name, or you could sort it by active ingredient, so that when someone asks you a question about whether something works, you have a place to go to quickly look at how many studies are there, what did they say in general. And then this is more recent. I've been working for a few years now on updating profiles of biocontrol agents. And this is an example about a predatory mite that can be found in orchards. And the link will be in your email as well. The goal here is to get practical information in a easily skimmable format. So we try to pull information that's relevant to a grower towards the top, like what crop do you use it in? Does it work? How well does it work? We're still building this website and getting more biocontrol agents up here. So by all means, if you see something that's missing that you know about and would like to write about, please let me know. I have a template. It's super easy to write these profiles, and I'm always looking for more authors. And that is everything I prepared. I'm happy to talk about some more questions. My contact information is here if you want to reach out, and also the link for my blog, which I think I put in the email too. So that's what I have for today.
Thank you so much. We do have some time for questions. If anybody, maybe I'll give a quick moment if anybody has a question before I jump in with all my questions.
Well, I got one, but it's your turn, Janet, not my turn.
Fair enough. I guess my question, so I work with a lot of like very conventional 8th generation growers who have been spraying the same things for a long time. And I've been really surprised that recently when we have like advisory committee meetings and generally asking for feedback, a lot of people have been sort of throwing this at me of they're interested in trying to dabble in biopesticides, trying to dabble in biocontrol, sort of which I think is hopeful. I think it's interesting. I think a lot of it comes from regulatory or marketing standpoints. But I guess that's just the preamble that my question would be for somebody who doesn't have any experience at all with this kind of product and is interested in trying to dabble in it. Do you have any recommendations of how I could help them to make it a successful first experience?
That is a great question. I'm hoping that what Jaime and Kerik have to share might help address this, because I think directing them towards products that there's more data on, like where it's more likely that this is going to be effective, is probably one option. Something that we've already worked out, like here's exactly how you should use it, and it's probably going to work. I think the other piece is to help them get information about a lot of those details that I include on the biopesticide profiles, like making sure they understand how it needs to be stored, or other little things about tank mixing. One of the things I always ask with the bacterial biopesticides, bacteria in the active ingredient, not targeting bacteria, is about copper sensitivity, right? So that they're not mixing a bacterial biopesticide with copper in a tank and killing their bacteria. So I think helping them find the answers to those questions so that they're increasing their chances of being successful when they use a product.
Awesome, thank you. Just check out your website is the answer. And
well, I only have 11 profiles up there so far.
Mm-hmm.
If there's a product that you're really dying to see a profile about, let me know and I can bump it up the priority list.
Okay, awesome. Thank you. Any other, I guess you had one, Glenn, unless anybody else has anything.
All right, well, I will take advantage of the opportunity then. So I was talking here's an example of why it matters, these distinctions. I was talking to Quan Zhang at the New England Veg Fruit meetings, and he had said when you're doing for blossom protect against fire blight, and I'd like to hear what Kerik thinks about this. He said you want to put the blossom protect on first, and then use oxidate. So if you're going to do a 1-2 punch there. And I was talking to him afterward, and I said, well, that seems wrong to me because the oxidate is going to, it could blast apart your blossom protect fungus. And so I thought you wouldn't want to do that. You want to put the oxidate on first. And he told me, no, blossom protect does not act as a competitive antagonist. It doesn't, I thought it used up resources on the flower stigma and therefore denied those resources to the fire blight bacteria. And so you want to keep this living thing going. And then, you know, so the oxidate would interrupt with that. He said, no, blossom protect acts as an SAR inducer. It does not acting as a competitive antagonist. So these kind of details, it may sound, you know, arcane, but it really affects how you would use these materials effectively.
Glenn,
Yeah.
I can just talk about that during our part. That's very nuanced.
Well, but I think your point is correct. You know, some of these details make a big difference, whether it's mode of action or you know, something about shelf life or storage temperature. Or, I mean, there are a few biopesticides that say store in a freezer, you know, and how many growers have a freezer that's appropriate to store pesticides in. That's a legitimate consideration.
Any final questions for Amara? Otherwise, we might keep going. Are you going to be on till the end, Amara?
Yep, I'll hang around.
Perfect. Thank you so much. Great presentation. So our next speaker today will be Jaime Piñero, who will be talking about specifically about entomopathogenic nematodes and transitioning to on-farm trials and implementation. So this is some of the work that he's been doing for quite a while. And Jaime Piñero researches insect-plant interactions and helps develop sustainable fruit pest management solutions, such as attract and kill technology and these entomopathogenic nematodes. And I'm really excited to hear how this work is continuing to go for you.
Good afternoon. Thank you, Janet, and thank you for your presentation, Amara. It was very nice.
Yeah.
So
One
today,
final, sorry, Jaime, one
Yes,
final
Laura.
thing. There's like a block, a rectangle on the top right there. Perfect.
it's gone now.
Oh no, actually it's back.
Okay, let me just then hide it. What about that?
It's actually worse now. Oh,
Worse?
I have no idea what's happening, but now there's a little rectangle in the middle of the screen. There, perfect. Okay, don't touch anything. Go.
Okay, then yeah, I have only 15 slides because my goal is to engage in a discussion, hopefully. So I also want to acknowledge that research on EPNs and plum curculio specifically has been done for almost 40 years. I think the first paper was published in 1983, but only for the last 15 years, there has been more interest in this topic from the more applications for growers. So I will be discussing mostly plum curculio and at the end, apple maggot fly. And the question I want to pose is, why are we studying entomopathogenic nematodes, given that it's difficult for growers to believe that they work? Because it's very hard to prove that in the field. When you apply, then what? So you're not going to see reduction in injury in the fruit on that year. Maybe the population can decrease a bit the following year, but it's very difficult to do, I think, work in the field to convince growers that it works. And the reason why I wanted to include this slide is because in November 23, I did an online survey. It was about 10, 12 questions. I only have one to show you. So the question number 5, it was this. So we know that you can use entomopathogenic nematodes to kill insects and is effective at killing plum curculio larvae in the soil. Are you interested in this area or biological control agents? So we received 44 responses. And what you will see in the graph now, what you can see is that 52% are interested, they need more information. 41%, they already think they're effective and they would like to use them. When you combine those two with the green, one person already applying nematodes but on their own, that's 95.5% of people interested in nematodes. I think that explains, or that's the reason why we need to continue with the research, because people believe in nematodes and they're willing to evaluate them in the field or in their farms. So I have a 2.5 minute video because I really think that this is the best way to provide an overview of nematodes. So bear with me for 2.5 minutes. Can you hear that?
Entomopathogenic nematodes, also known as roundworms, can parasitize a wide range of insect pests. There are various species and strains, all with great differences in efficacy. The nematode Heterohaptitis bacteriophora is used to control black vine weevil. After application, the nematodes actively search for their prey and penetrate them via mouth, anus, or respiratory openings. Once inside, the nematodes feed on the contents of the prey, excreting specific bacteria as they do so. These bacteria spread inside the insect and convert host tissue into products that the nematodes can easily ingest. The insect dies within a few days. Affected insects turn yellow to light brown and become slimy or dehydrated. The growth of the population of nematodes within the host depends on two factors: the number of infectious nematodes that penetrate the host insect and the size of the host. The hermaphrodites can lay up to 200 eggs. The female lays her eggs inside the host, or when there is insufficient food, the eggs will develop inside the female nematode. From the eggs that remain viable, hatch larvae that rapidly develop. Several thousand nematodes can eventually leave the host. They immediately start searching for new prey. Curious about other beneficials?
Well, that will be the commercial at the end. So what you saw at the end, the smallest nematodes are the infected juveniles, and the mothers, the reproductives, are the bigger ones. So I really enjoyed this video because it's short and very clear for teaching and also for extension. If you go to Copper website, they have maybe 15 or 20 videos on other nematodes plus parasitic wasps, etc. They make very good videos. What I have now is a very short video that is specific to Pluncurculio, and it was recorded by a student. I only want to show you one thing in this. So the student is dissecting the Pluncurculio larva. The application of nematodes was about 2 or 3 weeks before that time. So what you will see is not the adults that infected the larva. What you will see is the reproductives already developed. Pull out. We just got there. That ball has hundreds, I don't know, perhaps thousands of nematodes which are ready to attack the next host. My point here is that that's one of the benefits of nematodes, that they can reproduce soil and attack more hosts. So this is specific to Pluncurculio, and the species was Sternernema riobrave. I'm not going to spend time on this because half of you know that we have been trying to develop this system for the last 15 years, is trying to kill adults, adult Pluncurculio adults in the canopies by using lures. You can attract them, concentrate the adults and the injury. You can kill them with insecticides. You have larvae infesting the apples, some of those apples. And what happens is that you have a density of Pluncurculio larvae under these canopies, which is greater than anywhere else in the orchard. That would be a very good way to apply nematodes in the soil, in like a targeted applications, because we have shown over time or multiple studies that you can kill up to 90% of the larvae in the soil. When it comes to field performance, this paper that you can see in the screen, it was published in 2020, and it was done in collaboration with all these researchers. The work was done in New Hampshire, in Massachusetts, and in Vermont, starting in 2016. I was away from here, but Tracy was leading this work. So I came back in 2018 to UMass, and we continue working together and trying to develop this. What you can see in this slide is that the number one species, the most effective, is Stenernema riobrave. It's a cruiser. That means that the infected juveniles are going to they're going to search for the host. But then we found that Stenernema carpocapse is equally effective. The problem with the riobrave species is that you cannot find it. I mean, it's hard to find it. You can go to Arbic Organics. Sometimes they have it, sometimes not. And I believe that carpocapse is more versatile and it can stay longer in the field, but it's not a cruiser. It's going to be more ambusher, just waiting for the larvae to cruise or to be in close proximity. We have also tested Feltii, the species Feltii, and it has been proven not to be effective. Here is a table. This table shows you from that paper is about 5 years of research showing in different conditions that every time you have riobrave, it's going to lead the emergence of adults to zero, very close to zero. But when you have just the water control, then you have emergence of adults. Of course, they're not being killed by the water. So every year is the same, but in 2019 is when we compared the 3 different species that I discussed before, and the one that was not effective, it was Feltii. You can see the emergence of adult plant curculio is the same as water, but riobrave and carpocapse, they were equally effective. Where can you apply the nematodes in the orchard? That will be the next question. Well, what I'm showing in the screen is this approach where you have these lures deployed within the canopies in the perimeter row trees, some perimeter row trees. You can congregate injury and you could apply nematodes there. But this is based on this research also, showing in blue, when you bait a canopy, a tree canopy with this attractive lure, you can increase the injury by plant curculio. That also means also larvae, number of larvae, by 3 to 10 times compared to the orange, which is any other tree in the perimeter. So you are aggregating the curculios and the injury. And what you can see in the overall is just the average of those 6 years of research. And those 6 years, it was done at 7 farms. So we had 42 data sets showing that, yes, you can increase injury consistently. And it doesn't matter which year. Every year will be similar result, that there is a significant increase in injury. Or on average, is 12% injury. So the reason why I'm showing to you these results is because how can you apply nematodes? A second thing that I want to discuss briefly is that what you can see in the top chart, A, in blue, that will be the injury on the trap tree, close to the average. The average in orange, anywhere in the perimeter, is going to be less than 2% average. When you go to the tree, which is adjacent, so left and right, there is a little more injury, but it's not comparable to the trap tree. I'm talking about B. You can see adjacent. But as you move away from those trees, injury is going to decrease a bit. So there is very low spillover effect. Of course, you don't have that effect in the orange because those are any trees in the perimeter. It doesn't matter. It's going to be a tree and the adjacent or behind. Like two away means behind the trap tree, 2 trees or 3 trees away. Then actually on top is just in the perimeter, 2 away, 3 away. In the B, the second chart is going to be the one that is 1 behind, 2 behind, or 3 behind into the interior. So very low spillover effect, which means that if you are using trap trees, you could target those trees for the application of nematodes, spot spray. But if you have a perimeter that is facing the woods, and we know there is more injury just in general, you could just go for the perimeter application. And I will tell you some information about the cost in a moment. Any comments or questions about this? The timing. For research, we have been applying nematodes in early July because this is the chart that shows the life cycle of plant curculios. You need to apply the nematodes when you have most of the larvae in the soil, not too early and not too late. So again, in Massachusetts, it has been about early July, the first week of July, and I think we have found very good results with this application. So don't go to early June or to August because it's going to be too late. How to apply nematodes. I also have another video to show you, but hopefully it's going to be early morning when there is more moisture, low temperatures in the soil. Hopefully it's going to be cloudy. Hopefully it's not going to be too hot on that particular day. Hopefully there is no drought during that week because we had that situation before. We had to add water. And then that's when it comes to how to apply it. But next is a video that it was recorded by Liz Garofalo 4 or 5 years ago. And she speaks very fast, but she did a great job in just 1 minute. Bear with me.
In a previous video, we discussed field-scale research on entomopathogenic nematodes that has been conducted since 2013. That research
how the
has
application
demonstrated
is
applying
made.
EPNs can result in an
35
85%
acres.
reduction on average of the plum curculio summer population when compared to the application of water as a control. The next phase of this work involves on-farm demonstrations. Research technician Jim Kripa demonstrates the application of the EPNs in one apple block at the UMass Cold Spring Orchard on July 17, 2020. 1 billion nematodes were applied against plum curculio larvae and pupae in the soil. The timing of application is important as we need to have most of the plum curculio larvae already in the soil. As part of this demonstration, 2 commercial orchards received 1 billion EPNs each. All EPNs were donated by BASF.
That's it. So this is a summary of what Liz Garofalo said. So I'm not going to discuss it. I can send the PDF and you can read it. Basically, low pressure. You can remove the nozzles of the tank. So again, I will just go to the next slide, but not the low temperatures. I mean, not during a hot day is recommended. But this is what was done specifically for that application. When it comes to the economics, I can spend more time here. It depends on where you buy the nematodes. For my research, some comparisons have been made with using Arctic organics and materials, different species, different formulations. So you can see in the instructions when you go to the website that when you buy 5 million nematodes, the recommended area to be treated is 1,600 square feet. If you buy 1.5 billion, you can apply 30 acres. Here is the price that we pay for those nematodes. And pretty much it's the same price for any species. There is also what is called a combo. You can buy a combo of 3 nematode species that is going to be a little more expensive. I think it's it's $72. So it's going to be cheaper to buy the combo than any one individually. So I would like to bring your attention to the 5 million and to the 50 million for one reason. For example, if you apply nematodes in the perimeter, like a spot application only to the trap trees, those that are baited with the lure, or just the perimeter, either way. Let's say you have a Macintosh tree that it has this diameter in the canopy. That means that the canopy area will be 116 square feet. Well, the recommendation, which I call the low rate, it has been 250,000 infective journals per square meter. So based on this area, you will need, so we're going to need to apply about 2.5 million. It's almost 2.7 million, but I can go for 2.5 million nematodes per tree. So if you buy the 50 million package, you pay $72 plus shipping expenses. And that will be enough, based on these calculations, to apply to 20 trees. It could be one perimeter, it could be 20 trees spot application around the block. Anyway, you can cover pretty well 20 trees, and that means that the cost is going to be $3.6 per tree. Is that expensive? We're showing, or also Dr. Shapiro, Ilan, that they're effective. So you can trust that they're going to kill the larvae in the soil, most of the larvae, and the cost will be $3.6 per tree. Then, I don't want to speak too fast, I'm going to get fast, but when it comes to apple maggot fly, there has been some research done in the past, but this is more recent, 2020 publications with Dr. David Shapiro. Mohamed Usman he spent time in the lab of David Shapiro he was focusing on this research. He is now at Michigan State University doing a PhD. And the plan is that Mohamed will come to my lab by the end of next year, hopefully early next year, to do a postdoc working on nematodes. So what you can see on the left is the initial evaluation, different strains, different species against apple maggot larvae and pupae. And what the group found is that Rio Brave and Carpocapse are the two most promising species for apple maggot fly. Well, what a coincidence. Those are the two best species for plum curculio. Well, the second paper on the right is also combining nematodes with fungi. I know it's a different topic, but the work is happening now. And they found that Rio Brave and Carpocapse, again, they're the most effective species. But now you could combine one of them, the most promising was Rio Brave, with an entomopathogenic fungus, Metarrhizium species. So that was done in the greenhouse, but no work has been done in the field. And this is the title of one grant that it was submitted early this year and is funded, actually it was last year, is funded. And there is 4 objectives. 3 of them is about extension, the trap tree and the monitoring, etc. But one objective is about research. And so the objective number 3 is going to be focusing on native entomopathogenic nematodes, also native entomopathogenic fungi in the soil. And this work is in relation to what we have been doing for the last 7 years, which is multi-cultivar grafting, because you can use the lures to concentrate injury by plum curculio. But as you know, no one is using the lures for that purpose. Then we decided to use cultivars, which are very attractive to the pest. Now you can use red astrakhan and other cultivars, graft multiple trees in the perimeter. Those become the trap trees, like a trap cropping. So the idea will be, what are you finding? What can we find in the soil, leading to, hopefully, over time, suppressive soils? So can you rely on not native species compared to applications of commercial nematodes? And the second question that we would like to ask for this research objective is, when you apply, for example, Carpocapse against plum curculio in July, single applications, as you could see in the video, in the second video by my student, there is reproduction. So they reproduce. Can you apply nematodes against plum curculio in early July? And then can you show that the same nematodes that you applied, hopefully, are they killing apple maggot fly larvae and pupae in the soil in August or September? So basically trying to connect 2 different pests and controlling those 2 pests with a single application of nematodes. That's the goal of the research objective for this article. And I'm done. I don't know if you have any questions or comments. It was very brief because I wanted to see if you wanted to discuss.
Great, thanks, Jaime. Yeah, we do have time for questions. I have a really quick question, which is that video, the copper video that you showed, could you put a link to that in an email or in the chat? That was really cool. I'd love to show that in the chat.
Yeah, I wanted to say that I will send you the link, because those videos are really, really cool. And I want everyone to, if they want to use them for teaching and extension, I will send you the link. Thank you.
Perfect, thank you. Any other questions for Jaime?
So Jaime, you showed the cost per tree, but if you just take like a 5-acre block or maybe even less, if you're doing a perimeter only treatment with the nematodes, so now you've reduced the sprayed area to, I don't know what percentage, what is the cost per acre spread over that 5-acre block? And would that perimeter treatment with the nematodes give you adequate control?
Yes, that's a good question. Then let me just go back to my slide, because if you apply the 5 acres, then the perimeter, maybe the area, maybe 1 acre, like a 20%. Then if you want to apply 1 acre, you could go for 50 million. So you could apply the perimeter row with about $75. the four perimeters, which it will be probably 1 acre, let's say.
You're saying, sorry, the price you're quoting us there, that's for the nematodes, or is that for the nematodes plus the spray? I guess the spray, it would depend what you're gonna use for that spray, but that's kind of the model you're working with, right? Is you put on the perimeter nematode application with those trap trees, and then you would still spray those trees with an insecticide. Am I understanding that correctly?
Well, the spray with insecticide will happen before, because you're killing the Bronchorculio adults. When you apply the nematodes, which is going to be early July, you will not have to apply any insecticides to kill anything. I mean, Bronchorculio, just the nematodes. So they will be applied in early July. I would say 50 million nematodes, $72 plus shipping cost, let's say $100. That will cover 1 acre. So if your perimeter is 1 acre, that will be the cost, but no insecticides.
So you're saying the following year, you would not need any Plumbaculio insecticide because of those nematodes?
No, I didn't say that. No, I will never say that, Glenn. No, what I wanted to say is on the same year, there is no need to apply, of course, never insecticides against Bronchorculio in July. That's what I was trying to say. The following year, I will still do the petal fold spray, the whole block. And then I will still go to the perimeter sprays for the adults. And then it's just, that's the difficult part, to show to what extent you are reducing population, because the adults are coming from other places as well. So you are killing the larvae and pupae of Bronchorculio that it will emerge otherwise in the summer. And those Bronchorculio adults that emerge, they are not going to reproduce in the northern strain area. They are going to be feeding. So there may be sometimes feeding injury to the apple or other crops just by feeding. You can reduce that if we can show it. But the following year, you can have a reduced population overwintering, but that following year, there are more coming from other places. So I don't think it's going to be possible to show. Maybe over a 10-year period, there is a way to show that there is a decrease injury over time, but that's the difficult part to show.
Well, that's my skepticism. It's because you're already done spraying insecticides for Plumbaculio for that season. So this is just an added expense because you're going to have to spray again next year anyway. So yeah, I mean, it's always good to reduce the population, but like you said, you really don't know how much you're reducing the overall population for next year. And that late summer feeding really isn't consequential. So killing the larvae in the soil isn't going to make much difference in terms of the belt feeding later in the summer.
That will be up to the grower to decide if they feel it's cost effective or not. I would say maybe organic growers would be more interested in this because there is no good way to kill the adults the following year. So anything that helps for organic growers, for example, may be very useful. But yes, if I could just show you that every year you reduce population by half and there is less injury and you don't have to spray petal fold, that would be a big promise. I mean,
do we have that data, Jaime, or is that something you're hoping to get?
What was the question?
Do you have that kind of data yet,
No.
where you applied in 1 year and then the next year you reduced feeding by X%?
No, I don't have the data. I
Okay.
have data when it comes to the grafted trees, but we have not applied nematodes every year. So I don't have that data.
Gotcha.
Final questions for Jaime? Yeah, I think that it'll be interesting to see where this goes going forwards.
I would like to address again Glenn's question, because at the end I said that maybe that will be something that we can show, that you can bridge maybe Bronchorculio and apple maggot. So if one application of against Bronchorculio can suppress apple maggot, then for the same cost, you are controlling or reducing the population of 2 different pests. Right? If I can show that in the field with the research in the next 3 years, 4 years, then growers may be more inclined to use them, because it's not just Bronchorculio, it will be also apple maggot fly.
Jaime, for the apple maggot, would you need to treat the entire orchard floor, or would you still do it as sort of a trap tree? Do you think you'd get enough larval control of the apple maggot to reduce those
I don't-
populations to reduce the spray, or are they gonna be flying in from outside the orchard? I don't have a good understanding of their biology enough. I think that's gonna be
Yeah, for
the,
apple maggot, I mean, for apple maggot, if you have a very clean orchard that they spray consistently, a good control, the population will come from outside. So I will say the perimeter will be effective. If you have an orchard that is very small, and then the control of apple maggot was not very good in previous years, then you have a resident population, larvae and pupa in the soil every year. I will have to apply to the whole block.
gotcha, okay.
I mean, I think too, if we can get NRCS on board to support this practice, then that kind of changes the equation substantially, you know, for growers to implement and see how well this works over 2-3 years. Do they see a decrease in pressure? And
And we can show that if you saw the cover slide, the first slide, did you see some emergence cages in the field? That's how we measure the emergence, using some overwintering cages. So we can do some of that work, but it's really hard to do like a large scale assessments.
I think something really important, and I know you have something along this lines already, but maybe the research is still ongoing, but I think it would be really helpful to have a concrete sort of best management practice using entomopathogenic nematodes for growers, sort of like a very clear and concrete kind of what we just talked about, but in handout format. You spray, put up these trap trees, spray those with an insecticide at this timing, put out the entomopathogenic nematodes at this timing, put them in these locations, something like that we can pass along to the growers.
That is a great advice. And with this grant, as I mentioned before, one objective is research. I discussed the research. 3 objectives is extension. So what I want to do is discussing with all of you interested in this area, see how we can develop those practices and the outreach to reach out to more growers so they can implement nematodes. There is an opportunity to get free materials, like a free nematodes sometimes. I can contact BSF and they can send me, I hope it was done before, like a boxes with 1.5 billion nematodes that I can split and give to growers. That can be done because that's extension work. So basically either or both research and extension, I would like to work with all of you if you're interested.
Jaime, you probably don't have an answer to this. I don't think the answer exists, but do you have a sense of what percentage of the plum curculio pressure is arising from within the orchard versus coming in from outside the orchard from the woods and other places?
Oof, if we have time, I can tell you because that was my dissertation for 5 years. I mean, 20 years ago. I can tell you, but by petal fall, you have about 60% of the plum curculios immigrating on average. It depends on the year, the conditions. Also, there is an effect of weed management. One of my chapters was about studying overwintering and to what extent they can overwinter in the orchard versus going to the forest, depending on the weed management in the fall. So if you have in the fall a weedy block, they may stay in the block. So we found that the information, and they're going to be still the majority will spend the winter in the forest, but a proportion will stay in the block. When you have a very clean area under the canopy of the trees, they are not going to be spending the winter so much compared to you have a weed area. But that also depends on the tree size. In the spring, when they're overwintering and they're becoming active, if you have large trees, they're going to stay more in the perimeter. That's when you have the perimeter row sprays most effective. But if you have very small trees, like a high density tree blocks, then they're going to continue to penetrate into the block. So there is multiple considerations, the with pressure in the fall, the tree size, but also what we found is that when the bloom is ending, based on captures in panel traps, remember the sticky traps, the sticky cards, the sticky panels that I was using, there is so much activity by plum curculios. There is like a back and forth orchard versus and going to the orchard, back to the forest, back to the orchard. So it's very difficult to record immigration, but there is so much activity by plum curculio, like a movement, just when bloom is ending, just before petal fall. I don't know if I answered your question, but
No, that was my sense of it, that
Yeah.
most of them are gonna be coming in from outside the orchard, so
Yes, most of them,
the
yes.
in-orchard population may not change the overall equation.
Right, I think we should
Yeah,
probably move along to Kerik's talk. Thank you so much, Jaime, and your
you.
information, I believe, was on that last slide. Everybody knows how to get in touch with you anyways.
Mhm.
Unless anybody, I saw you had your hand up a while ago, Amara. I did want to just give you
Oh,
space. in
no,
case you
I
had a
think
question.
my question was sort of related to Glenn's about, is this something you do every year and how often you do applications? So I'm all set.
Gotcha.
Thanks.
Okay, wonderful,
Thank you.
amazing. All right, well, thank you so much, Jaime, and we're gonna move along to the last section of this presentation, or this question session. So Kerik Cox will now discuss some data from biofungicide efficacy trials, along with information regarding the cost of these products. Kerik Cox wears all the different hats and seems to do a little bit of everything. His research focuses on fruit pathology, fungicides, and fungicide alternatives, along with a lot of work on stakeholder education. So your screen is up, looks great, and you can take it away.
Move any potential dark boxes away from everything. It seems like we should be going good. All right, yep, lots of slides. Won't talk about them all. These things were developed by myself, and I think for today's stuff, my co-author is probably Mackenzie Sheshel, putting together a lot of these materials for me and doing all the trials. She even has a project on Blossom Protect, Glenn's favorite. So I think because there was a question about that, I'm going to focus more on fire blight. Now, we don't need to talk about a lot of what I've discussed because you've already discussed what these things are. Amara's presentation was absolutely wonderful. I have stuff on modes of action or not, and some stuff on summaries of modes of action, but we don't want to worry about that. What I want to talk to you first is about succeeding with biopesticides. And I don't know for apple growers or vegetable growers or small fruit growers, but I showed these slides to small fruit growers on Long Island because the best way to make these things work that I found over the years, as I've done them, I've actually made them successful at Cornell orchards, and it may be so successful to the point where I get comments from other scientists like, I don't believe the data, other than the fact that I have 15 different people taking the data over multiple years, and we still always get the same results. So how to beat this? I think to make these things best is start with horticulture. Use the best horticultural practices. Have the best farm that you can. It needs to look like you would be willing to eat food off the orchard floor. So in the world of apples, and you could apply this to whether you're growing currants, I've seen them also look in the same fashion, is tall spindle apple orchards great, but you can see immediately that this is just going to be a haven of biological control failure. Yeah, even though the apples have bags on them, because these are two shots I showed in China, it shows a tall spindle semi-dwarf. You need to move away from this and towards this, because these better well-spaced plantings, there's just one on the right, the tall spindle is, let's switch to the laser pointer, is actually producing more money per acre than the semi-dwarf on the right. And you're getting much better color, you're getting better yield, air circulation and drying time. I often say the best fungicide in your planting is air. Exactly. If you can move air through this, you're doing great. And you might say, well, these things are very young and vigorous. That's another problem with fire blight. Yeah, this could survive fire blight, but this is going to have a pantheon of other issues, insects and diseases as well. And you're going to get more bang for your buck with that one. Great air circulation. What else I often say, if you want to win, I think you're at a stage where you have to be very agile with disease forecasting tools. And I don't care which one you use, depending on who you talk to. One is always better than the others. I use this one, and we could show data, but we're going to switch and talk about fire blight on how these different things work. It doesn't matter if you're using any disease forecasting tool, you're better off. They're going to tell you which conditions that are predicting pathogen maturity, dispersal, and infection. And just like your traffic predictions and regular weather predictions, these are only accurate to a point. They can't predict the most finest biology, and the most finest, it's going to be 3 drops of rain at 3 o'clock. Any service whether it's your traffic system or your weather app or your disease forecasting that's telling you they can predict with great accuracy precipitation or the presence of water, I would be immediately skeptical. Or minute, very detailed things about the pathogen biology, such as the Canidia, are all germinating right now and they're all mature. Once they get too detailed, I tend to be a little skeptical. But for the most part, these things are great. Now it's becoming more challenging to use these tools, and we have workshops on how to deal with the nuances of one, such as in the ones in NUA. We have Ask Us for Maturity. This is the apple scab model, talks about infection events. And with each complaint from other extension specialists, the models just get better with time. So keep telling us they're wrong, we'll get them better. And the problem and the trick with a lot of these things is that now climate change leads to erratic weather. When these things were created, it was like potted plants and mist chambers getting infected with apple scab fungi. It's not like someone came around after hours and started flipping the temperature up and down and hosing the plants down with water erratically. And so the models, regardless of which one you use, were never developed with erratic precipitation and heat and cooling in them. Do we need to make new ones that account for this? I don't even know how one would do it, other than to literally set up the experiments and in the middle insert a bunch of erratic weather. And those are things that some of my colleagues in food science and I have talked about doing and related to fungicide persistence. But anyway, what they generally seem to be very good at is increasing the precision and effectiveness of management applications over a basic calendar schedule. If your operation is massive, you might just have to do the calendar schedule and spray more often. If you're a small farmer and it's farming is hopefully your full-time job, you might have the agility to do these things. If you're running a research, teaching, and administration program, it's very hard to do these things, but we do it. Otherwise, we wouldn't continue to get grant money. So you're often spraying in weird times of the day when the weather says it's time to go, more so than when someone says you're allowed to do it. They're pretty cool. I use the NUA system. I use RimPro. I use I use Cougar Blight. I use them all. They're all telling me pretty darn good things. I haven't, even my weather app on my phone, it's pretty all right, all things considered. And the nice thing I like about NUA is you can have a local station, and if you don't, or a rampaging deer or apathetic teenager smashes your weather station, it will still pull in leaf wetness algorithms and stuff like that from satellite data, making it a pretty nice tool for even just monitoring the weather. And it does work in forecast mode as well, which is pretty great. So one of the reasons why is there so much interest in biologicals, a couple of different reasons. Consumers think that biopesticides are safer and tastier and don't persist on fruit. The regulatory agencies also agree, as do many countries and in Europe. And a lot of the reasons that we were trying to move away from multi-site fungicides, the big ones, the captan, sulfur, the mancozeb, the coppers, yep, even coppers, is they tend to reduce soil microbial activity. They are reproductive and developmental toxins to animals, and they have probable human carcinogens, and then there's synergistic toxic effects with insecticides on pollinators. The EU was like, ugh, these things aren't good for mammals, birds, aquatic, bees, and insects. I was working with my friend Christoph and looking at fungicide persistence and detection, and I was like, we should look at this one. And he's like, he looked at the chemical structure. I didn't even tell him what it was, and he's like, oh, we're never spraying this ever again. Do you see what these rings mean? Do you see this double bond? That will kill us. I was like, oh, that was mancozeb. And so anyway, these are the reasons we're moving away from some of these things. And I embarked upon an entire set of research that we're going to skip today. And you can see many other things, and the EPA is removing them, but that's something I'll talk about next week at Janet's meeting. And I wanted to start thinking about, could I use biopesticides to get rid of multi-site fungicides? And we're going to skip this, and I'm going to go talk about fire blight. But you can just imagine all these different topics I could talk about, reams and reams of data that we could show. And you're more than welcome to ask questions about that later. But I thought in this case, since Glenn received some grief over the use of Blossom Protect, that we should talk about some use of biopesticides. And this is where they work really well, in my opinion. They work pretty okay on fungi diseases if you use them right. And I'll talk about how to best use them when I talk about spraying them, maybe in the next slide or so. But fire blight is a great bacterium. It's sitting on the surface of the flower. It will kill your entire orchard very quickly, yes. But it's out there, it's exposed to the environment for a brief period of time, and toothpaste and water will kill fire blight. So many of these other things will work really well. And so I'll be showing a lot of data, different kinds of products. And this orchard site, that's kind of like my larger vertical axis planting from China, except I've spaced the rows out so far and the trees so far apart. And you're like, Kerik, why on earth would you do such a thing? No grower would ever do this. And I'm trying to fend off the review trolls from peer-reviewed published manuscripts. If my treatments are too close to each other, they'll say that each material interfered with one another. So that's why everything is so strangely widely spaced. And so, boop. So I always showing some blossom blight trials, and we'll let that blossom blight go into shoot blight in general. And what I tend to do for my biopesticides, including systemic acquired resistance inducers, and Amara's right, acylobenzo S-methyl is not necessarily a biopesticide. So we put some of these stimulants, defense inducers in there as well. We'll usually go pre-bloom a little bit to build up the theory of populations. But in my case, the biopesticides that kill, we'll call them, whether they're like a fermented soup that makes an antimicrobial metabolite, I'm going to put these on right before I inoculate. If there's something that needs to build up a response, like a defense inducer, they may go on a little bit earlier, 20 to 40, but the killing things and antibiotics that I have in there for standards, they're going to go on right at 80% bloom. And then I'm going to come back and I'm going to dump a dose of fire blight on, and that would be inhumanly possible to generate in an orchard other than by directly smearing ooze of fire blight on a flower. A really hard test, because you're going to want to see how these things perform under hard conditions, because when fire blight gets going, things get hard pretty quickly. And I'll just be looking at the types of data you're going to see are the incidence of blossom blight, percentages of blighted shoots, blighted flowers, 5 or 6 plot reps. If it's a high density plot, and we're starting to move into high density fire blight trials as well, but I think you're mostly going to see that vertical axis orchard. And they really behave very similarly between the two orchard styles in this situation, other than you kill the high density plot if you let it get away, and you'll never recover very well. But that's a different talk on how to manage fire blight. But this is going to let you see how to do it. Over the years, I found that when you're using biopesticides, the best way to be successful is when you're mixing them and when you're looking at your equipment and calibrating everything. You need to pretend like you're filming a video for social media where everyone is going to criticize everything you do. You need to be that fastidious. It's no longer a matter of putting the material in the tank, looking at all the residue on the container, and throwing it over your shoulder. You need to like quintuple rinse. You need to check your nozzles. You need to make sure your equipment is going to be putting it out and that everything leaves the tank at the end. It requires a level of fastidious that one would only employ if they knew an audience of the internet was watching them and ready to criticize at any moment. I find that when I take that level of care, you definitely don't want to hire a spray person that throws it over the shoulder. You want to avoid that altogether. These things need to be used with the utmost care. They're also very expensive, as we'll talk about a little bit later. And so that's when I put these things on. It's as if I've got someone scrutinizing me, the DEC on one side and then an internet troll on the other. That seems to be the key to success, I find. So for shoot blight, for the most part, I have done inoculated shoot blight trials, but it really involves smearing fire blight, concentrated fire blight on recently wounded shoots, making it really hard for anything to work. But what we like to do in many instances is look at the natural development of shoot blight from the blossom blight. And we do put materials on. Now, a lot of them are defense inducers, because at this point, fire blight's inside the tree and is killing from the inside. And you can dump everything you want on the surface. It's only going to manage surface materials. But if it's inside the tree, then nothing is on the surface, and you need to attack fire blight from the inside. So we'll do things like defense inducers like ActiGuard, LifeGuard, and Regalia, biopesticide type components of defense inducers. And these are going to need to go on prior We have a lot of work on PGRs, such as the gibberellic acid inhibitor Apogee as well. And then these are the types of things you're going to work. And you're like, is Apogee a biopesticide? No, neither is Actigard, but they're excellent tools that work well in a conventional orchard. At least on the eastern United States. They don't work on the West Coast, I'm told. Anyway, so we'll look at it. We'll look at the progression or look at the number of blighted shoots on the tree. Oh, there's 20% of every first-year shoot is on this dead. You know, in that particular instance, and those are the types of graphs you'll see. So we'll start a little bit, and I'll show you some different materials. These are what things look like, lots of different products. And once again, what we're looking at is the percentage of blighted flowers. So in this particular instance, I protect the flower, I inoculate the flower, I come back with protectant again, because these things do direct kill and can give you very limited, next day, we'll call it limited, level of kickback material. What do we have? The non-treated, I can kill almost 90% of the flowers on a tree with this inoculum dose, which is a very strong test right there. The shoot blight, you can kill about 70% of all the shoots on a tree. Really devastating. And we have a lot of different products. The product always, Firewall is the antibiotic standard. It's streptomycin plus a PGR Apogee at 6 ounce. This is sort of the silver bullet for you wanting to control fire blight conventionally. And then we have a variety of different things. And I've kind of color coded them based on what I thought they used to be. Other than this Eco Swing is in the wrong color now. That's fine. But for the most part, green things are like usually fermented microbes and other organisms. So what double nickel is the bacterium. I've made it slightly pale blue because there's a little bit of copper in there. That's Quava. Howler, another microbial fermentation product. Thea, another one as well. Different types of bacterium that are souped out. Agrophage is a virus that kills bacteria. You'd be like, wow, look at that big bar. The larger the bar in any one of these graphs that you see, the more variable that performance of that product is between plots. So Glenn could use it in his orchard and someone else could, and it can be like a smashing success, or it can be a catastrophic failure. And some trees are a catastrophic failure, some trees are a smashing success. Hence that variability that I like to show. Actinovate, that's another, I think it's an actinomyces type material, another microbe. Eco Swing, I can't remember what this one is off the top of my head. Blossom Protect, it was already discussed previously, but it's been alternated with Provisto. Maybe it needs to get the blow, but it doesn't exist on this particular one. Thyme Guard is essential oil. Thyme. Alum is a pickling salt, like you would literally pickle your pickles with and buy from McCormick Seasonings. And Cinerate is another essential oil. These types of things down here are in the grass products, generally regarded as safe by FDA. Use them as food additives, and you can kill your fire blight with them. For the most part, what I've really noticed pretty well, I've really enjoyed using the Howler and Thea products. I love double nickel LC because it's a liquid and it mixes well. When in doubt, pick a liquid. Those work really well. And of course, Blossom Protect works fantastically, and it knocks down so much blossom blight that you don't get a lot of shoot blight. So now that you've kind of seen what this looks like, I'll just sort of move through different seasons. Oh, if it's a really bad year, the weather is so much, you have basically an environmental catastrophe of fire blight. It doesn't matter how many sprays of streptomycin come through if there's a volcano of fire blight flooding through the orchard. And you can see this in this season that nearly killed every apple orchard at Cornell AgriTech, regardless of what they were. Mine tend to survive better because my population is streptomycin sensitive, because I intentionally inoculate my blocks with a strep sensitive isolate. You can see, like, wow, even streptomycin's pulling 20% that year. It has a pretty good luck with the pickling salt as well. Blossom Protect, a little weaker in this really tough year. And you could see that once the infections get going at about 20 and 30%, nothing stopping shoot blight. And even Blossom Protect sort of fell apart here. We have Oxidate, formerly mentioned as well, a dangerous combination of acids and hydrogen peroxides that will blind you and burn the skin off your body. But by all means, let's use it because it's environmentally safe. I don't like using that one because sometimes birds nest in your orchard. So it's also be mindful to know which ones will actually physically harm you. so that you don't hurt other wildlife that may be just trying to find refuge in your plot. Did pretty well. So these are on populations, and this is sort of alluded to a little bit what Quan Zhang was talking about, like in support of his idea, is that wait a minute, wait, this is the populations of the flower strep good, blossom protect, the populations of fire blight in the blossom are huge. That's terrifying. Some of these essential oils can really work and knock down flower populations out and oxidate as it's burning you and birds in your orchard and other creatures that live there is also burning out flower populations. We take a look at what the things look like for shoot blight. Pretty rough year. It was really hard to keep it under control. And the trends kind of mirror blossom blight. We had a pretty good year with that alum stuff becoming pretty popular. And even though there's a big failure of controlling populations, we see a good bit of blossom protect. And we can answer Glenn's question towards the end. Let's skip through a couple different ones. Then we had a cold season. Great. Everything looks pretty good except for an old formulation of Serenade that's a liquid. And you can use essential oils to kind of beat down the blossom blight. On a really cold year, strep works great. In a really cold year, blossom protect looks great. That agrophage is cool and it works, but it's very variable. And I don't know how to make it less variable. The virus has to be fed. And as we all know from COVID, you can leave your groceries on the front porch and the virus will die. So in that case, if we're using a virus to stop fire blight, if I leave it on the leaf in the middle of the sun, yep, that's right, the virus will die. And maybe in some cases it was more shaded and better. Once again, I looked at blossom populations. Yeah, it's terrible. Blossom Protect is not removing the populations. They're accumulating inside the flowers. And look, I even have a stray bee floating really high up there. That's kind of cool. Others just completely eradicated them when it's really, really cold. Alum, centerate, oxidate. I mean, we all know that cinnamon oil can hurt you. You've seen the videos of people putting sugared cinnamon in their mouth and choking and nearly dying for popularity on the internet. So imagine this happening to fire blight when you blast it with cinnamon oil as well. 2023, shoot blight, great. You control blossom blight. You can control a lot of your shoot blight as long as the weather remains cool in this cool, dry season. Couple other weird things. We might stop to see if we have any other interesting products at different years and different blocks. The shoot blight. I don't do all of the flower colonies in some of these. What else do we have in this one? Fireline. You can see one of the things I'll talk about if you ever see a general fire blight talk is I'm not a big fan of oxytetracycline as an antibiotic. And you'd be like, oh, is that oxytetracycline, that Fireline? Yeah, it's quite variable. I mean, you know, you're doing better with Synaction for the most part. And now we have things in color-coded. The greens are your antimicrobials. Lifeguard and Apogee is a combo that was popular. But you can see with a bigger bar, more variability in there. And the more inconsistency it's going to work in your orchard from spot to spot, place to place, year to year. The antibiotics, very tight. So things like, well, double nickel quaila, it's maybe not the best, but it's got a tight bar. Probably because it's a liquid and flowing completely out of my sprayer in that particular instance. I've been really excited. The Howler did not work that well. It did. Howler works really well on fungal diseases for a completely different reason. You'll have to see a different talk for an explanation of that. And then there's a lot of interest now in going after fire blight with copper. And this is the 2022 hot, wet season. Everything died. And so that's why the bars and everything is pretty rough for some of these interesting copper products. I do like the Magnabond. Why do I like Magnabond? Because when I put it in the sprayer, it's all on the trees at the end. Why do I like Kirazen? It's a really low dose of copper that's combined with zinc to enhance its ability. I feel like at this point, we might need zinc in our apple trees if we're allowed to use in any way, shape, or form for various leaf diseases later on. When else we talk about that? It was a cold year. Everything looks golden for these extra types of weirdos that we have in 2023 and some of my assorted stuff. All of them equally effective. Error bars are very tight. And you're like, well, it seems like the success in beating fire blight with biopesticides is have a cold year. Yeah, that will do it. Or a not cold Memorial Day, or whenever the silly things go into petal fall. I did do a couple blossom, looks like I did do some blossom populations in here. Things look pretty good. Once again, you can see that we're not removing, this is another form of blossom protect. We're not removing the populations of the fire blight bacterium. Yeah, it's down a little bit, sure, but not as much as all these weird coppers and when we have some coppers in there, and it looks like this is mostly just coppers and a sporicide type fungicide as well. And you can say, oh, well, look at that. Pretty good. A cold year means great work. What can we sort of wrap up with? Let you ask a bunch of questions. I've been pretty happy with Blossom Protect. It's highly effective. And you're like, wow, is it doing it? Why is it doing it? And why did Quan make that comment to me at that meeting? Howler and Theo are looking really good. They were now bought by Certus. I recommend them for other reasons. Alum, we have a big national thing on fire blight biopesticides. Working pretty good, particularly for me and Washington State. The defense inducers, whether they're systemic acquired resistance inducers, ISR inducers, I get pretty good performance with Lifeguard and Regalia, particularly in combination with Apogee. Actigard can work fine as well. It's also nice, but I use it differently in the early season. I don't really talk much about that. I'm pretty excited about these new products that I've been testing for a couple of years. Low MCE copper, what that means, metallic equivalent copper. Low dose of copper enhanced with zinc to remove the killing part. Well, I was happy to see that it did murder the fire blight bacteria. Let's talk about economics, because this is what you really have came to see. So one of the topics that I talked about for is rotating. I like it when you can rotate the biopesticides in with bigger, heavier materials. It doesn't add extra labor. And you can, if you use disease forecasting, you can reduce your costs by only applying these things when the going gets tough and not every Monday morning. So you can save a lot with this. I do a lot of berry talks where we're showing like you can get the same level of control with half or less than half of the sprays. So rotation is good. There's also a practice of taking a biopesticide and mixing it with something at risk for resistance. When you do that, the labor and the cost goes up, which is pretty frustrating. Now, biopesticides are not necessarily more expensive than single-site fungicides and high-quality Cap 10, Mancozeb, premium liquid formulations, the kind of liquid formulations that don't damage your crop because they're so well made. I think for the most part, if you have a lot of acreage, these prices are going to be cheaper. If you're a regular old person like myself, buying them from a distributor directly, they're going to be around $35 to $40 USD per application per acre. What is the cheapest? So sulfur, sodium bicarbonates, minerals, oils seem to be the least and are the most. I need to fix this. Are the most inexpensive. They're not the least. They don't want too many double negatives in there. Yeah, those are that's sort of the most inexpensive products. When you start having to ferment things, store them in your refrigerator, as Amara already mentioned, or they have really fancy premium formulations, they get really expensive. And for the most part, when I'm asked to test them, I don't actually often get asked to test them with adjuvants compared to the single-site products. So in that case, you're not necessarily paying for the additional adjuvant as well. Other last stuff as well, be very careful. You might think Captain's cheap. I'm just going to do it all the time. Injury on specialty crops, whether it's apples, berries, or whatever, very high, particularly in tank mixes. They get complicated because you are going to be putting out entomopathogenic nematodes and other horticultural materials as well as insecticides. When the drying gets slow, Cap 10 really can hurt things a lot. This is another really popular idea, is mixing two products and spraying the same amount of time. That will greatly increase the cost, even if you are getting fungicide resistant management. 35 to 45 here, put that with 35 to 45 DMI fungicide, and you've got an $85 fungicide or disease spray only. Yeah. What could you do? I've heard a couple of people talking about the idea, if I'm just going to go alone, I think you got to go with increased frequency. Go with every NUA predicted infection period, maybe up to twice a week. Could increase the cost greatly, but make it more successful. I'm going to go ahead and let you ask as many questions as you want, so I can, and as long as you want to stay. Please don't ask many questions.
Great, thank you. So there was a couple that already came into the chat one. Amara was asking about specifically with Thea, but just in general about whether these products might be harmful to bees and how much of a current concern that might be.
Yeah, for the most part, this is I in the slide set that I will provide to Janet, you can see I've got all kinds of other, there's stuff about specific ones. For the most part, probably not, but I wouldn't go out and spray anything that has an SDS that requires you to wear eye protection during the middle of the day. I mean, clearly in this beautiful picture of Blossom Protect known as Botector for blueberry and mummyberry, and there's like bees all over the place flying around in there and doing stuff. Stuff that tends to harm, powdery mildew is usually kind of caustic in nature, like oils. I mean, I wouldn't go out there and spray my beehive with CMS stylet oil or ride the air blast right by and clog them all out. Or if they're flying, I wouldn't do it. So I think stuff that would hurt you, it hurts. Yeah, I got it on myself at one point. Don't tell Cornell. Yeah, I think those could be harmful. They do not have an environmental persistence that would harm the bees, I think, is where people get excited. Next question.
Maybe the takeaway upshot is just don't spray your bees as much as you possibly can avoid.
Yeah,
But that they maybe
evening
don't
is
have the
great.
residual.
And I will make another plug for evening sprays because a lot of these other materials will break down in UV light. Oh, it's feeding in UV degradation with agrophage. So if you spray them at night, you don't get a lot of UV light. You're laughing all the way to greater increased efficacy.
Awesome. You already answered this question, but there was a question about sharing your slides. So yeah, if you just send those to me, I'll put it in the follow-up email.
Yep.
A lot of information. So I think everybody needs more time to process it. Mike, did you want to unmute and ask your question, or do you want me to say it for you?
Yeah, I can ask. For tank mixing, Kerik, usually at Blue, we're doing something for fire blight, but then also getting a scab spray on. Does the fungicide basically destroy the Blossom Protect, or?
All right, now that's a great
mix?
question. We've done 2 years now in young high-density orchards, putting Blossom Protect out and literally intentionally spraying with fungicides. My student and lab manager Mackenzie has a lot of slides on this. I think we showed a few at Eastern New York last year. Basically, what ends up happening, there are a few products that do harm it. Captan and I mean, a lot. Not to name specific names, but these are specific fungicides we tested. We didn't have an impact for Maravon, but we found that the fluopyram and Luna Sensation seemed to impact populations. Now, that sounds terrible, but I'm saying impact for the day. These silly things built populations back up on those flower surfaces like within less than 24 hours. They're just maxed out. And last year in the wet season, Mackenzie was very sad. It's like, I saw no differences amongst the fungicides. And I was like, great. That just tells the grower they can spray a scab spray with whatever the heck they want. And the experiment had 3 phases. Mix them together, put them on first, or put them on after the fungicide. And then mixing them together wasn't actually the worst or the most impactful way. But these things, this Aurea basidium is normally present on apples and will ruin your Sweet Tango apples. So this thing is a natural epiphyte that's just happy to be back in its natural environment. It's almost one of those cases where augmentation did not use an invasive organism to manage the disease. So kudos to the Botector Blossom Protect stuff.
All right, thanks.
A couple more coming in the chat. You guys can feel free to unmute yourselves and ask the questions directly yourself, or else I can read these out.
I'm going to save you.
Awesome. Thank you.
So, Kerik, I was really turned on by your comments about sprayer sanitation with some of these biorational products and just how astute an operator needs to be to have it clean to start with and absolutely sparkling clean after. And I get the question a lot, and I've joked that we're moving towards a warm tank, biological temperature with Ringer's solution in it, fully stainless, and dedicate it to something, right down to something like a nematode, where they'll just get chewed up by some of the pumps. Do you foresee a time where we may have dedicated sprayers for some of these more sensitive products?
Well, I would almost say yes, except for the fact that the EPA will remove everything but these. So in that case, we might have a dedicated sprayer for some of these things. I don't know if I would mix, oxidate with any of my natural product type, but maybe clean the tanks out with them. I wonder about that. Maybe not in the same extent that we would have a herbicide sprayer or a stone fruit sprayer. I'm going to go spray my azoxystrobin for my peaches and then drive right into the McIntosh orchard to make my, and to defoliate all the trees.
Yeah, that changes your day.
But I think we need to be more cognizant. I wish we had a new Andrew Landers that would come around and yell at everybody for nozzle calibration, checking things with spray cards, because I think that's going to be the key. The problem is, like with everybody, I'm taking the kids to Taekwondo and then I'm running over to UV the orchards at night and trying to get back in time to pick them up. And that's where my YouTube level of like fastidious can fall apart as we're scrambling to get something done before fire blight or something kills the orchard. And so I think maybe making sure, like you mentioned, ahead of time, is that if, you know, whoever's doing it, you want the most type A person possible.
Cool. Thanks.
And to your point there, Kerik, I do think to our credit, we have a few folks who are doing those sprayer calibration, sprayer safeties. I think those are super important. I know Jason is doing some up in Ontario. I've been trying to get him down here, but the logistics haven't worked out. Mike, remind us, you have somebody doing that in Eastern New York, right? Or maybe Mike's. Anyways, yeah, we do have some of that going on, but absolutely, if anybody's willing to help us out with like a little more of those calibration workshops, I think those are amazing. And unfortunately, I started just around the time that Andrew Landers ended, so I haven't gone through one myself, unfortunately. George Hamilton. Jeremy had a question also. I guess maybe I would more broadly break that down as like with strep resistance or without strep resistance. I know you have some really good articles on this, Kerik. How would you recommend alternating? Something like Blossom Protect, looking at the NEIPM. I don't know if you want to jump in and add to that, Jeremy.
Yeah, no, I mean, just Kerik, thanks very much for your presentation. And yeah, just looking at, all right, if we have those weeks at points where, you know, we have multiple infection events, sometimes they're wicked high EIP values, and then maybe it drops down and we're at like, I don't know, 80, you know, like, is that the time to think about? Yeah.
So watch when I get to the NUA forecasting and how to do it and all that stuff, when you're over 100 on the EIP scale or about 200 on the Cougar Blight, which works just as well, despite the fact that it looks less complicated, that's when you want to go with the biggest. And I would be tempted to tell you, if you want to do the antibiotics, do them for the first big one and the last one. It often seems that now what we're getting is we finally get some of our cold bloom, but at paddle fall, my colleague Ken Johnson has found that the infections can actually be almost worse than they would be even at full bloom or early on. The reason I say maybe try the antibiotics early is because if you can reduce the initial inoculum, you're going in with a curve that will be shifted towards we call it terminal bud set in the fire blight world, so that you don't end up killing the tree. So you can shift the curve that way. And it's kind of like putting on your urea for apple scab. You're taking that initial all source of fire blight bacterium and flattening it so that when it does try to kill you later on, there's not as many of them there. Fire blight's kind of like a numbers game at some level, and the fewer infected blossoms means fewer infected shoots. You probably noticed that the trends seem to mirror. So even though you can get a huge outbreak of shoot blight later on, completely unrelated to blossom blight, yeah, I think doing it then and then hitting it strong at paddle fall. In the case of strep resistance, if things get bad, it's time to save your crop and worry less about having a fresh market crop. So when that 2022 year hit, the entire AgriTech became strep resistance, including the USDA germplasm repository. And I'm like, do not spray any antibiotics. And they have been keeping that place alive with just blossom protecting copper. And that's what I've been telling people. It works just as well. And it doesn't work just as well in my trials. It works just as well in George Sundin's, Ken Johnson, California, Tiana DuPont. We're all getting success. I would be more worried maybe if you're more like Pennsylvania, you're going to need something to murder stuff. But you can use your oxidate in a manner that was recommended by Guanzhang. Yeah. I hope that answers it. But we just go blossom protect, and I wouldn't even use antibiotics anymore. I feel comfortable enough with this product that I would use it. And even though it takes 3 years and thousands of dollars more to plant an orchard at Cornell because of all the rules, I'd still feel comfortable just going blossom protect.
All right. Thank you.
That's with copper or just Blossom Protect by itself?
Just Blossom Protect by itself. Sometimes if you see ooze, that's when you might want to put the copper out to keep those ooze droplets from rubbing against each other on the wire and on other shoots. Yep. Yeah. Copper is to keep the surface junk down if you see ooze, and Blossom Protect is just and that only goes to bloom. Once you're after petal fall, do not do the Blossom Protect and only go copper for your summer.
High praise there. Amazing. Thank you, Kerik. We're
That's good stuff.
gonna call this the end because we're already
Great.
a little over time. If anybody has further follow-up questions for Kerik, I think y'all know how to get in touch with him.
Yep.
We will be sending out that email with Kerik's slides with some of those links from Amara. And obviously, if you have follow-up questions, feel free to contact any of our speakers. I think Jaime had to jump off, unfortunately, but hopefully y'all know how to find him as well.
And,
Thank you guys, everybody. Thank you to all the presenters, to everybody who showed up. And just as a final reminder, we won't have a session next week. So the next session will be 2 weeks from today on February 13th. So I will see you all then.
And we'll
Bye.
be talking about ambrosia beetles, so we'll see you
yes.
there.
I
you.
love ambrosia beetles.
It'll be fun. We'll see you there. Bye, everyone.